Hello everybody, thanks for joining the Agrigenomics session. The demand for high quality nutritious food is increasing. So is the need for environmentally sustainable, safe and clean methods to grow food. The agriculture industry is adopting new technologies like the next generation sequencing to address this increasing demand and the safety norms. Next generation sequencing is an advanced technology that needs technical expertise to understand the complex workflows. Especially, you need to understand the genomic complexities of the various plants and animals to be able to develop assays that are specific and sensitive for the breeding programs. And the amount of data that is generated on an NGS platform is humongous. You need to have bioinformatics expertise to understand the data and use the various pipelines to interpret and use it in the breeding programs. So that makes it very challenging for the agri industry to adapt technologies like the NGS. At Thermo Fisher Scientific, we take pride in our mission to enable our customers to make the world healthier, cleaner and safer. In line with our mission, we have been working with the agri industry for decades. As technology enablers and solution providers, we have multiple genotyping platforms like the PCR, Sanger sequencing, the Axiom microarrays, and the Iron Torrent NGS platform. However, depending on what is needed and the type of application that the customer is pursuing, the number of markers that they need to genotype is where we make the appropriate recommendation. For example, if one is doing early phase discovery work, trying to look through millions of SNPs or hundreds and thousands of genomic loci to pick and select and predict, we would recommend the Axiom microarray platform. Similarly, if you're in seed QC or trying to do animal parentage verification, you would be using the quantitative PCR, the Sanger sequencing and the capillary electrophoresis platforms. However, once you have decided or identified on the relevant markers that are important and crucial for developing your new breeds and where you really need to scale up your testing, you would like to scale down and identify a mid-density range platform so that you could scale up your testing. That is where the AgriBusiness team has adopted the Iron Torrent technology and the AmpliSeq chemistry to develop a mid-density platform called the AgriSeq Targeted GBS solution that can help the customer genotype for known targets in a mid-density range of 50 to 5,000 markers. And the AmpliSeq chemistry helps in developing a multiplexing strategy where you can look at all the targets, amplify all of them in one single reaction for coming up with the most appropriate solution for marker-assisted selection or breeding that the agri industry can use for furthering or meeting the demands. The AgriSeq solution includes a service that our team provides in developing custom panels for the known targets. We work with the customers in identifying the genomes of their interest and develop these panels. Further, the AgriSeq library prep kit, which is an adaptation and modification of the AmpliSeq chemistry to scale up and to make it affordable. Likewise, we have developed about 1,536 barcodes that enable high throughput genotyping. We use the Iron Torrent Gene Studio S5 Prime and the Chef for sequencing and make use of the Torrent Suite server along with other additional pipelines to provide the data analysis. And combining with the various sample prep solutions that we have, like the MagMax Agrigenomic Core DNA isolation kits, we have a complete solution that you can trust, thereby can move forward with a total solution that could help you for your affordable breeding programs. Coming to the AgriSeq workflow, it can be easily divided into 4 steps, starting with library construction, templating, sequencing, and data analysis. For library construction, we start with 10 nanograms of DNA as the input. With the AgriSeq library kit, the whole protocol is a 6 to 7 hours protocol, which can be performed both manually and automated. We do have a 96 well format that works in both, and a 384 well format that can be fully automated. So once you have amplified your targets, prepared the libraries, and barcoded them, those amplified barcoded libraries are cleaned up and normalized, and then added to the Iron Chef along with the templating reagents. For templating, that happens overnight. And the next day morning, you arrive and take out the chip out of the Iron Chef and load onto your sequencer for the sequencing. No matter how many samples or how large is your panel, the sequencing always takes only about 2.5 hours. And so in a given day, you can easily sequence 2 chips. In the templating process, the Iron Chef is actually able to template 2 chips. Therefore, your sequencing is also set up for where you can sequence 2 chips in a single day, which gives you a throughput of up to 3,000 samples in one single day. And the data analysis follows the sequencing, and it usually takes about 6 to 24 hours, depending on the number of samples and the size of the panel, to convert all the raw signals and give you the actionable genotypes. And that is all seamlessly integrated. So overall, the workflow is pretty simple and mostly is automated. And you can easily scale it up by adding automation. One single full-time person can actually genotype up to 3,000 samples using a liquid handler, one sequencer, and a chef. They can easily process 3,000 samples in a day. Starting from the sample to output, the whole workflow is about 3 days to accomplish the AgriSeq workflow solution. The data analysis from sequencing is pretty seamlessly integrated. The Gene Studio system is where all the sequencing is happening and the raw signals are recorded. The Iron Torrent server runs the Torrent Suite software and all the plugins. It's a cloud-based platform that helps you run. In the Iron Torrent platform, the basic technology is that as every time a nucleotide is incorporated according to the template, you are releasing a hydrogen ion is released as a nucleotide is incorporated. And this release of hydrogen ion causes pH changes and changes in voltage, which is what is recorded. And once all these raw signals are processed and linked to what base was incorporated corresponding to the time frame and the change in the voltage meter, is how you convert the raw signals into the base calls. Once the templates are sequenced, all these bases are then aligned to the reference genome of interest that was being studied, and the actual calls are made. And using plugins like the coverage analysis and the Torrent variant caller plugin, which is actually looking at the presence or absence of the variant at your hotspot or marker of interest, those calls are recorded. And our team has developed the AgriSum toolkit, which helps you summarize all of these calls and presents it or helps you visualize and makes it your quick analysis or review of the data very easy. And also further reformats this data into a usable format that is very helpful for your downstream analysis. So at the end, you get actionable genotypes that you can transmit or take it over to any of your downstream analysis tools that can help you process your data. So here's the sample output from the AgriSum toolkit. It summarizes the data. As you can see here, it gives you the panel summary on the left. It tells you what markers were in the panel, how much was the overall coverage and overall mean call rate. It also provides you a quick preview into how the different markers in your panel performed and how the samples that you've tested perform. Our team has used the simple traffic colors, the green, yellow, and red. Green meaning good, yellow is be watchful, and red is something has not worked out, go check it out. And as you can see, there are drop downs that will actually indicate which of these markers or samples failed in the runs. You further also get to see nice graphs and charts, gives you a quick preview of how the sequencing run was and how the performance was. You can also get graphs where you are looking at multiple parameters. In this example here, you can look how was the mean depth or coverage versus the call rate. So you get an idea. Did the sequencing run happen as you anticipated? And was the performance of the markers looking good, or do you really need to go repeat this experiment? It kind of gives you a quick preview into your data. And at the end of it, the AgriSum toolkit really facilitates the various formats in which you need the data. The default format is genotype matrix, and then we've also giving formats like the top bottom, which is similar to the microarray format, and the parentage format that one would use for doing parentage analysis. The AgriSum toolkit actually makes the whole data interpretation very easy and facilitates the using of the sequencing data for further downstream analysis. Looking at the Ion Gene Studio, some of the advantages is that it is easily scalable and flexible. In fact, here we are talking about the plant and animal genotyping, but the same Ion Torrent Gene Studio can be used for other Ion AmpliSeq applications for viral and bacterial typing, as the flavor of the current season is. The Gene Studio is being used to look at the COVID viruses, SARS-CoV viruses. You could do RNA-seq methodologies and various metagenomic applications as well on the same Gene Studio platform. But for us, for plant and animal breeders, the flexibility is also provided in the various type of chips that we offer, which kind of helps you with your throughput. We have the smallest chip, the Ion 520, which gives you up to 5 million reads, where you can test a few markers, say like one plate, up to a small panel of up to 400 markers, and about 125 markers and 400 samples. Whereas most of our agri breeding, agri customers who are into the marker-assisted breeding and selection programs use our high-throughput chips, the Ion 540 and the Ion 550, which gives them a range of 60 million to all the way to 130 million reads, where you can look at markers ranging from 100 all the way up to 5,000 markers. And depending on the panel size, you could always run anywhere between 96 all the way to 1536 samples. So it kind of gives you a great flexibility in coming up or optimizing your sequencing or genotyping workflows, which is what most of our customers would like to see. That's an added advantage with the platform. So overall, if you look at AgriSeq, it is a very flexible, powerful, and cost-effective tool, which was perfectly designed for agriculture, especially for helping our breeding customers. It is flexible because in one single panel, you could look at multiple types of markers like the SNPs, the multinucleotide polymorphisms, the small insertions and deletions, and other structural variants. You can develop assays that could include all of these markers. You can look at multi-species panels in the same run, where you actually would develop libraries or amplify libraries for each species, and then combine all those libraries and put them on for the sequencing on the same sequencing run. Similarly, it is very powerful because the entire workflow can be automated. It helps you in discovering novel genotypes because we use a 200 base pair amplicon. You are also looking at, oftentimes you are looking at novel genotypes that these torrent suite software automatically gives you, looking at the sequences that have been genotyped. It is very cost effective because in one single PCR reaction, you are able to multiplex for all the markers, whether it is 50 or 5,000 or even more. In one single reaction, you're performing a ultra high throughput PCR reaction. Similarly, you're able to genotype up to 1536 samples in one single run on a chip. And you can genotype up to 3,000 samples easily in a single day. Thereby, it is making the whole solution very cost effective. So once again, we can conclude that the AgriSeq is a flexible, powerful, and cost-effective tool that is been specially designed for agriculture applications. So here's some data that we can look at, which kind of supports our claims that AgriSeq produces high-quality genotyping data. We have looked at multiple size panels. The first one on the left, you can see the bovine 200 SNP marker panel, which is the ISAC recommended markers. And we have developed another panel for porcine with 1500 SNP panels for one of our customers. And in both cases, as you can see, the overall call rate and the performance of the panel is really high. 98% for bovine and 96% for porcine. We've also done several intra-runs and inter-run comparisons, and there again we see that the intra-run and inter-run concordances are pretty high, about 99% and above. We've also compared it with orthogonal technologies out there on the market. One of our partners tested our panel, our ISAC panel, and compared it with the data on the Illumina B chip, and we see 99.4% concordance between the two technologies. We further also looked at several plant panels working with our partner Eurofin Biodiagnostics. We had developed a panel for sunflower for about 768 SNPs. The samples were provided from the US grain repository, where they had samples with known genotypes. And so we compared the data between our lab and Eurofin's, and that is seen in the green and blue dots here, and then compared it with the data from which was provided by the grain repository. And we see a very high level of concordance between all the runs. So thereby we can say between sites and between runs, as well as with orthogonal technologies, the AgriSeq solution provides high quality data that you can trust. Here's another example where we had conducted a test for the USDA. We were testing out 19 bovine breeds that are used for beef, and we were testing samples using the ISAC bovine panel, and you can see that the panel performed really well. The average call rate was 98.5% across all the beef breeds that we tested. So overall, if you see, we have been continuously using innovative approaches to enable agriculture research. Over the past 5 years since we've launched this product, we have continuously increased the complex markers that we are looking at. The complexity has increased from SNPs to multinucleotide polymorphisms and insertions and deletions, and this year we are developing panels with structural variants. We have continuously evolved. We have increased the throughput from 384 barcodes to 1536 barcodes by the beginning of this year, thereby providing a high throughput option for our breeders. Likewise, We have been continuously working in improving our analysis tools, starting with the Torrent Suite software. As I have shown you data on how the AgriSum toolkit helps in data visualization and summarization. We have been working continuously support our customers. Overall, we have also launched several off-the-shelf panels like for ISAC bovine parentage. Last year in 2019, we launched the GBS panels for canine and feline parentage, and they can also be combined with traits and disorders. So we have off-the-shelf panels that customers can readily use, not only for parentage, but also for looking at various traits and genetic disorders and defects. In these past 4 years, our team has developed over 150 plus custom panels covering over 45 species in plant, animal, and aquaculture. So you can believe and work with our team. So overall, we are dedicated to continuous improvement. We have been developing various new applications and solutions for agriculture. As you can see, the various plant crops that we have worked on, including vegetables, nuts, and recently oil palm. Similarly, we have worked on livestock and companion animals and various aqua species. We believe in continuous improvement. We work with our customers in trying to work on as per their needs, whether including complex markers or coming up with faster design tools. We are helping them with workflow improvements by sharing automation scripts and coming up with the liquid handling protocols to help them automate the entire process. And we work very closely with various KOLs across the world in coming up with solutions that are helping in advancing agriculture. And we are always open to new collaborations and looking for more partners. Hopefully after this, the genetic analysis virtual conference, we will get more partners to work with across the world. And please do check our website for the various customer testimonials. As you can see, we have been working with great partners empowering global agriculture across the world. And we would like to see your name added here. Definitely check out for the customer testimonials, and there's a lot more useful information on our website. With that, I would like to take thank you for attending this talk and would like to take questions. Thank you.